that's called commensalism. All three of those are symbiotic relationships. We may see some commensalistic relationships in our studies, but the majority of them are going to be mutualism and parasitism. And your book will always, well, I shouldn't say always, but almost always try to give you an example of this, and an example of this, okay, to round off that information. So that's a strategy behind these labs. Okay, not only just, okay, we've got to memorize all these names and learn what they look like. Well, yeah, but that's probably the last thing you want to do. It's really not what we're excited about. We're excited about, do you understand how this works together? Okay, how everything works together. Okay, so today we have, I'm just going to write two words up here, just so I don't forget them. Today we have, in the bacteria, we have mostly working with the actual bacteria themselves, the little tiny bacteria. So we have an experiment. The first thing that you're going to do there is we have an experiment that allows you to gather some information about the way these things live, or especially where they're found, and how we might be able to understand how we can reduce their numbers. Not that they're all bad, of course, but there may be some situations where you could pick up a bad one, see? Or it could spoil your food or something. So you're going to do an experiment to show not only where they're found, but also what are some effective ways of reducing their numbers. Okay? So we have some plates here. They're grouped in twos. Okay, if you've never worked with a Petri plate before, okay. Oh, before I forget, because I keep forgetting this. If you're interested, if you're going into the medical field, then it might be worth your resume at least, or at least your benefit for contacts and other things. To look into the pre-med club that we have here on this campus, which is also something commonly found in universities and other institutions across the country. And UC Merced has a pre-med club that communicates with ours and do things together. And we get a lot of literature, a lot of invites to go to conferences and workshops and things like that through the pre-med club. Other organizations that are involved with training pre-med students, whether it be health allied or actual medical students, they all know, we have websites for the pre-med club. They all know that we exist, and they all solicit their services of conferences and things to help promote the medical field. Okay? And if you intend to go into the medical field and you don't attend any of those things and don't do any of those things, I can tell you there are other students doing it, and that doesn't look good for you. Okay, they are getting information that you are totally in the blank about. So if you're interested in the pre-med club, I believe they have a meeting at 1 o'clock this Friday, tomorrow, upstairs in room 227, I think. It's a conference room upstairs. Or if you can't make the meeting, you may want to drop by one of either myself or Carrie Coburn or Ms. Macias. We have applications that you can fill out and give us so that we can start the contact procedure. And the first meeting tomorrow is basically to determine whether to keep that as a time and place, or to change that, or to find another. We'll either meet upstairs or sometimes at the student union. But last semester we met upstairs. So if you are going into any kind of a medical or vet, which is medical actually, veterinarian, whatever, professional field, then you might want to think about looking into that, and at least for some time in your life, be a member of a pre-med club someplace, okay? So that it will show up on your resume or application someplace. Okay? You might be thinking about that. Anyway, I kept forgetting to remind you, and even though it's just the second week, tomorrow's your first week, okay? Reminded you. I think they're planning their next trip to SUMA conference, which is in Stanford, and it's a series of workshops and things, especially centered on helping students to know what it's like to get into medical school. What are the things you need to do? And it's run by students and faculty members. And so there's students of different levels, different beginning students and college students who are doing internship. So they hold a series of workshops, and they tell you about financial aid, and all that kind of stuff, and they show you how you can make deals so you don't have to pay upfront for your education, and maybe have part of your education, a sizable part of your education, forgiven under certain contracts. So they show you those tricks that people use to get through med school. So it's one of the better conferences that I've attended in the past. It's called SUMA, and it's centered to try to help underrepresented students who may not think they have the finances or background or things like that, to give it a step. This is what you need to do. It's not as, it's competitive, but it's probably not as impossible as you might think it might be. There's a lot of opportunities. So anyway, that's the kind of stuff. The field trip, the trip there is in February. So I think it's February 20th. And so those are the kinds of things that the pre-med club does. Okay? Questions? Good. And this is the prime group. Learn about that. So this is the Petri plate. Okay? Now it has a top and it has a bottom. You notice I have it upside down, because on the top is condensation, okay? And condensation is water, okay? And we don't want the water necessarily to drip into the agar, which is actually a product of another protist, seaweed, and actually is a food item that we eat, okay? There's desserts that are made out of agar, similar to Jell-O, but it's a little harder in consistency, and it's very, very popular in this country now, and in a lot of countries, okay? But they flavor it. But anyway, bacteria like to eat it too. So, while bacteria like to live on it because it's moist, it gives them a substrate, but then we can put in and choose what kind of nutrients to put in there so that the bacteria will be happy and can metabolize. We can grow. So, when you work with the Petri plate, you'll start, of course, with it on your desk, like this position. And when you're ready to inoculate, you need to use something that is not here. We'll find it. We'll get some out here. You need to use. Oh, here they are. You need to use a brand new pack. You need to use a swab. Okay, swab. And you need to feel where the little cotton is and where the stick is to open these. There, this is now opened. Okay, it's sterile. So when you're ready to inoculate, you would pull this out, and you would take a swab, and then open this up, keep it as an umbrella, and then swab your plate. Try to swab it forward. Okay. You might even swab it this way. Turn this as you're swabbing. You might turn this as you're swabbing. Okay. And then turn it as you're swabbing. Turn it as you're swabbing, and get all the bacteria off. you won't see them. So don't try to say, oh, I don't see any. Let me try to say, oh, no, I don't see them. Let me try to say, oh, no, I don't see them. You got them. Okay? If they're there, you got them. So that's sterile procedures. When you're done, break your stick and throw it away. Okay? So they're in pairs. We only have 6 groups. 1, 2, 3, 4, and you could probably get 6 groups in this. We've been doing 5 groups to do our other things by row. But if you want, you can probably divide yourself into 6. So I would say groups of 5 would be close, because I think there's 31 students. But groups of 5 would work pretty good. Well, there's a few absents. So those are good groups because there's 2 plates. 2 plates? Must be 4 plates. Well, yeah, there's 4 plates. I'll have to talk to the technician. What is this over here? That's something different. Let me talk to the technician. I think she underestimated. So just hold off on that for a second. We need 4 plates. And now we have half of them. So now we need 10 people in. Well, I'll talk to her. We must have more initially order. So now the other thing is that there's bacteria that don't live on anything to make the food necessarily. What they do is they make their own food. They have chlorophyll, and they photosynthesize. They were our first photosynthesizers, bacteria. Okay, and they photosynthesized and they produced sugars, just like a plant does. Okay, that's why when one of your questions asked which came first, photosynthetic organisms or eukaryotic organisms, well, bacteria are prokaryotic. Photosynthesis started in the prokaryotes. That's why photosynthesis is first. See? That's what that question was designed to do, is to get you ready for that. So we have 2 photosynthesizers here today. One, actually, that we want to study. Now remember, we're only working with, in this part, we're only working with prokaryotes or bacteria. Okay? So I'll tell you right now, this is not a bacteria. Okay? This is a vascular plant, even though it's small. Okay? How small? Okay, this is a vascular plant, okay, right here on my finger. This is a zola. It's a fern. It's a fern. And it lives floating on the water. We don't care about azola, okay? We really don't. We're not studying azola today. But we're using azola only to help us to understand symbiosis. Because in azola, the fern, it has bacteria. Nostoc lives inside. Cyanobacteria lives inside azola. Okay? So we're more interested in the symbiotic relationship. Now, true, ferns can photosynthesize. Okay? So what is azola getting out of this? If it can already photosynthesize and it's using a photosynthesizer, well, it couldn't be photosynthesis that it's looking for, is it? Well, azola, since it lives on the water, sometimes the nutrients are a little shabby. And one of the most important nutrients is nitrogen, because nitrogen makes amino acids. Amino acids makes proteins. Proteins makes enzymes. Okay? And the only thing the DNA ever codes for is proteins. So nitrogen is extremely important growth product. Okay? So azola is relying on nostoc to nitrogen fix. That means nostoc takes atmospheric nitrogen and makes a nitrate out of it. Okay? Makes a nitrate. Because nitrate is what we use for fertilizer for plants. That's what they need. That's the form they need. Okay? So the bacteria does that for azolla. Okay? Now, you may get a question about how we might be using azolla and cut the cost of rice. So rice is not so expensive. Rice is a very important product. It feeds more than half of our world's population. Okay? It's very important. So, these things grow in the water. Well, if you know anything about rice, rice grows in rice paddies, which contain a sizable amount of water. And these things float on there. Okay? And so when the rice is ready to harvest, they go ahead and harvest the rice, pick them out, pick the little grains and stuff. And then they can drain the paddy, and azolla will get stuck in the mud. And then since there's lots of nitrogen fixed in the azolla, then you churn that into the soil, and now you have ready-made fertilizer. You didn't go out and buy it, you didn't go out and make it, you didn't borrow against it, nothing. It just grew there on its own, because it's a photosynthesizer, azolla, and it's utilizing nostoc. Nostoc is also a photosynthesizer, but it's more interested in getting some of the plant products that azolla is making. So it has an easy, it needs some kind of some nutrients, such as the minerals and things that are picked up by the azolla. So nostoc uses some stuff from azolla. Okay. So if you're answering a question about that, make sure you emphasize the nostoc, nitrogen fixation, and the fertilizer aspect of it, because that gives us more information. Don't spend a paragraph talking about azolla and what a wonderful ferment it is. And don't call azolla a bacteria, okay? That's been done too. Okay, so that's a little information about azolla. We'll see if we can get it right this semester, okay? Anyway, we also look at bacteria to understand a little bit how we classify bacteria. They're classified by their shape, okay? Okay. And in order to classify bacteria, shape is important, and we have three shapes that we look at. Okay, one is a rod, one is a sphere, and one is a spiral. Sometimes a spiral just looks like that. And there's names for these. Okay, there's names for these different ones. This is called a sphinx. That's the rock. This is called a caucus. I can't write that. I don't know why. And this is called a spirilla. Those are the names of the shape. Okay? Sometimes they'll use it as the name of the genus too, but it's the shape. So basically, this exercise is to have you pick up the slide and observe the different shapes. It says three forms. There's separate smears. And a gram stain. I'll talk about gram stain in a minute. So anyway, you're supposed to find the three forms. I'm not sure, I don't remember what this slide looks like, but it's, oh, I see. There's a smear on the top, a smear in the middle, and a smear on the bottom. I just couldn't see the smears looking this way. So where's the three smears? So there's three smears, okay? And then one smear will have caucus, one smear will have. So you want to make sure that you draw those in your pictures there, and you can label them, identify them, size them, okay? Size the skin. Not them, the skin. One of the ways that we can see them is we use a gram stain. Okay? And there's a chart there that you fill out. Bacterial species gram reaction. So is it positive or negative? Okay. Who knows anything about gram staining? Okay. Well, Graham is the name of a person. Basically, the stain contains two stains. I won't list them because you're not, maybe you can make one. if you ever take a microtype class, you will get the names of the stains, but I don't think the stains are, this is just more information for you. But there's a blue or purple stain. I'll just use the word blue here just to keep it simple. And there's a reddish stain. Now you really only need the blue stain, basically. The blue stain, if you have an organism, and I should tell you, there's two huge categories of organisms. One has a certain kind of cell wall that will take up the blue stain. It has chemistry in it that attracts the blue stain. And the other bacteria has more lipid stuff in its cell wall, doesn't attract the blue stain as well. So it won't stain at all, okay? So the ones that stain with the blue stain is called gram-positive, okay? This is called positive. But because we still want to see the bacteria, we have to apply another stain to show up the ones that don't take up the blue stain. So the ones that take up the red stain are the gram-negatives, okay? And so in your slides, you'll see sort of a purplish bacterium or a reddish, pinkish type bacterium. And those are the result of the stains that we're using. Okay, so the purplish, bluish are the positive, gram-positive, the other one's gram-negative. And how are they important? Well, it's because of the cell walls and because antibiotics work on the cell walls. Doctors have categories of bacteria that they have assigned antibiotics that work on certain kinds of bacteria. For example, penicillium, okay, or penicillin, works on, is a wonder drug, practically. It works on all kinds of bacteria. Okay? And what's another one? Erythromycin, tetracycline. Those are all different types of antibiotics. So the doctor's going to try to pinpoint the type of antibiotic that works on the specific type or category of bacteria. They don't want to kill all your bacteria, and they don't want to give you something that you don't need. And some people can't take some antibiotics. Some people are allergic to penicillin, so they have to modify it and go to other things. So the positive and negative helps the doctors to figure out what kind of antibiotic to use that's most effective. Okay. Questions on that? To give you an example, E. coli. Right? What do you know about E. coli? Escherichia. Escherichia coli. What do you know about E. coli? Right, it's part of your gut flora. Okay? So the doctor, if you have a gram-positive infection, the doctor is going to try to find a gram-positive antibiotic because it doesn't want to kill E. coli. That would be devastating. That's where you get all your B vitamins, most of your B vitamins, and you get your vitamin K, which is blood clot. So the doctor really doesn't want to make more of a problem here. So, and the main thing is that you get B12 is a very, very important vitamin, and it's difficult to absorb in many people, through the stomach at least, and parts of your intestine. Intestine is a little easier though. But E. coli that's one of its favorite things it likes to make, is B12. So you go to the doctor and you you're allergic to penicillin. So the doctor might say, well, you need to take this instead. But it's going to kill your E. coli too. So I need to give you a B12 injection, and then you need to take these B12 tablets, okay, under the tongue, so it absorbs. So they're going to modify that, knowing that, well, gee, I'm sorry, we're actually hurting you, but we're helping you. But we can help the hurt a little bit. So that's what you have to be careful with antibiotics. And that's why you don't share antibiotics. You don't say, oh, yeah, I got some left over. I didn't take all my antibiotics. You can take some of mine. You think you have a kidney infection or urinary infection? Here, take some of mine. Oh my God. If you only knew. Don't do that, okay, whatever you do, don't do that. Throw, if you have any left over, then you shouldn't. Okay? Well, maybe you got sick, and the doctor said, oh, go stop that. Or the doctor finds out, no, you don't have that. We just found out from the lab, you've got something else. Stop taking that, come in, you need to pick this up. You need to pick this other one up. So now you have a whole bottle of antibiotics. Throw it away. The best thing you can do is throw them away and forget about them. Okay? All right, that's the bacteria. That's most of the lab problem. The next thing that you're going to be doing is you're going to start studying some classification. On page 220, you start to learn some phyla. Okay? Phylum Euglenozoa, phylum Chrysophyta, phylum Dinoflagellata, phylum Rhodophyta, phylum Phaeophyta, phylum Chlorophyta, phylum Oomycotes, phylum Amoebozoa, and phylum Alveolata. Now these, well, this is one presentation of classification. So today we're going to look at the phylum Oomycota, page 220. If you notice, they actually give you phylum Stramenophylla, see? And then they give you Oomycotes, and they don't tell you what it is. Well, what it really is today, because these things change their position a little bit, the Stramenophylla, or Stramenophylla, is actually a clade. Now, we haven't talked about clades yet, okay? It's a clade name. Clade just means a group of similar organisms, with similar characters, okay? And then the actual phylum that your book really is intending to use, because they listed it on the top, is Oomycota, okay? And so that is in water mold. The water mold we have over here, and we have some living water mold, and we have some slides. Oh, this is your other blue-green algae, Oscillatoria. I wrote it on the board, but didn't say much about it. It's just a free-living one, as you can see. I'll have to grab the, or is the part with it? No, what is that? Okay. I'll have to grab the water molds. They didn't put them out. Actually, you saw them water mold already. You remember? Oh, is it cold? You actually saw them already. Okay. The actual inoculation that we have is the living water mold. Saprolignia is the genus. Your book will talk a lot about another one, I believe, but I'll tell you what the deal is on that. But we're doing Saprolignia, okay? That's the genus. And remember when you measured that little ball and you tried to get the diameter last in the microscope lab? That was the oocyst, okay, of the Saprolignia. It had its eggs in there. And the little balls were its, well, actually the big ball was called an oogonium. Okay, oo means egg. Okay. Oogonium, gonad, see, gonium is a container. Eum is a container. Okay, like sporangium is a container. Okay, so this is a egg container, oogonium, and it starts off with oocytes, egg cells. Once they're fertilized and they build a wall around each one of them, then they become an oocyst. See? After they're fertilized, the male, and they're listed in your book. The male sexual structure is called the antheridium. Maybe I should write them down. They're in your book, but it's all the time writing them. So the male, Saprolignia, the male sexual structure is called an antheridium. Okay, antheridium. what do elk and male deers have on their head? And you can use that to remember. And the antleridium, no, antheridium, but it sounds like it, right? So that's the male container. And in the saprophyta, it's just a growth, it just appears in the iridium. Okay? And these nuclei here are important nuclei. And at some point, it's going to form an oligonium. Oligonium. And it's got its oocytes in there, so you measure here. And this is going to grow a little fertilization tube, see? A little fertilization tube. And we'll put a little nucleus and then we'll fertilize the egg. See, one, this is going to fertilize this egg, and this is going to fertilize. And so that's how they grow. That's how they fertilize. That's sex. Because they want to fuse. Sex, sexual. Saprophyta also has another growth. It grows like a fungus. It has hyphae. It has little strands, clear fibers. It's nuclei. At some point, it's going to form a tube like this. And it's going to, by mitosis, see, by mitosis, it's going to divide off lots and lots and lots of little cells. And this is asexual. And that means these are not designed, these are just mitotically produced. Okay? And the word that they're using is they produce zoospores. Zoo, right? We have a flagellum, or a couple of flagella. And this is called a zoospore. Okay? Have you ever been to a zoo? Some of you have, but a zoo is where they keep animals, okay? Usually big animals that you can see pretty easily. So the word zoo, or zoa, means animal, animal-like, see? So these are like animals, because they swim. Okay? So in our live culture, we have smashed a cricket, and we've infected it with the water mold, and the water mold is producing some of these stages. If it's producing the asexual stage, it's releasing these little zoospores. The only way you can see zoospores is if your diaphragm is really low, and you see these little things swish by real quick, so like they're swimming protozoans or something. They're actually the zoospores. So you're trying to see, what you're trying to do is you're trying to see the life cycle of this thing. Trying to see the parts that make up the life cycle of the first real eukaryote. So that's a saprophyta. Saprophyta is a genus. So you underline it, capitalize that. All of these other words are not general, so you don't really have to capitalize them. You underline them really. So your exercise asks you to draw a zoospore angium. Zoospore angium. Zoospore angium. Angium means container. Angium, container. And or sometimes we use the Latin in the angium. So you say, well, that's a zoosporangium. So you're talking about one of them. If you see many, you say, oh, did you see all the zoosporangia? See? So in Latin, the is the plural, and the e is the singular. You know, you can go in a parking lot and you say, oh, look at all those cars. Doesn't that sound strange? You want to see all the car out there? Something's wrong there, obviously, right? It's just an S I didn't add. Same thing in science. When you start to speak science and you don't say, you say, oh, look at all these sporangium. It's the same thing, see? As me saying, oh, look at all those cars. Okay? So get used to writing singular and plural when you're talking, when you're writing, okay? Because it really, it's like someone taking their fingers on the blackboards that they used to have when you have science, okay? The next, the example they give you is they're asking you to look at Phytophthora. Phytophthora. That's another species of a water mold. The reason they talk about it is because Phytophthora infestans is the water mold that attacked the potatoes in Ireland in the 1800s, that caused an enormous famine and instigated a migration to the United States. Phytophthora infestans changed history for good. You'll find that in biology there are many organisms that have changed history, have won wars, lost wars for good. Okay? You'll find that biology is behind that. Egypt would not have been so successful, have been so successful if it wasn't for C4 photosynthesis. Remember C4 photosynthesis versus C3 photosynthesis? C4 photosynthesis is found in our grains, such as corn, rice, wheat, and Egypt cashed in on the wheat. Made a huge civilization. China cashed in on what C4 plant? Rice. Huge civilizations were established because of C4 plants. Changed the course of history. See? So, that's what I'm trying to impart to you, some of the importance of biology, rather than fun. I like history instead. Well, let's talk now. Okay, so the next one is amoeba. Yeah. Amoebozoa. So we have amoebas for you. Amoebas are single-celled eukaryotes, okay? And amoebas are fairly large. You can find them on Scanny, and then maybe go to 100 power. Or 400, but I think, oops, I'm silly. You don't have to go to 400, I think, because these are quite large. If you can't find it on the Scanning, then give up, don't look, or call me over. Say, can you find it? So don't say, oh, maybe I'll find them on 400. You won't. You'll find something, but it's one of those little dirt things you've been calling me over for, right? You don't want that. So you want to make sure you see these. And they won't look like they're moving at first. They look like a big, sort of darkish, granular blob of some sort, especially when you first pick them up and put them in there. They're more ball-like, and then they relax and they start to stretch out. They move by pseudopodia. They move by little false feet. They flow through their feet movement. So you might want to read a little bit about what that means. How does that locomotion take place? Because that's part of what we do here, learn the locomotion. Anyway, you want to observe the living one, say from this one here. To find them, you need to grab some of the stuff on the bottom. Scrape a little piece of the bottom. Don't scrape the whole bottom, because other people have to gather them too. Scrape a little bit off the bottom, suck them up. Quickly put that drop on your slide, because if you don't quickly put it on the slide and give them and you're sort of chatting around or looking at a cell phone or something, they're going to stick to your dropper, and then when you squirt the water out, they're going to be in your dropper, stuck to the side of your dropper. So you want to get them on the slide. See, so bottom and quick, and then and then cover slip. Okay. And we also have Amoeba proteus slides for those who want to see Amoeba proteus stained and shows some of the parts. And we have an Amoeba proteus model, which I'll bring in here. That's another. I got to get two things now. I got to get the model, and I got to get more more of these. And what's the next thing? Then we go to the next phylum, which is Alveolata. Okay, Alveolata is an organism. And actually the phylum itself is not. Alveolata is a clade. And we'll talk about that later in taxonomy. But Alveolata has a group in it called the complexans. Okay? And that's because they have a special organ on the top here. It's called an apical complex. And it allows this organism to successfully open up the cell membrane and get inside a cell of their host. Okay? And the one that we use is the Plasmodium. And can you see what genus we have, or what species? Is it Plasmodium vivax?
Yeah.
Yeah.
And this is the causative agent of malaria. Okay? Malaria. Malaria is the disease that a human has. And the mosquito, as you learned from the quiz, is something that carries that disease organism to its next host. It doesn't have the disease. The mosquito doesn't have malaria, but it imparts the organism to something else, to us, or another mammal, or a bird, and it will infect that. Now there's many species of Plasmodium. There's Plasmodium falciparum, Plasmodium japonicum, there's Plasmodium vivax, and I've got a little book that shows you the Plasmodium stuff. So the Plasmodium, there's a life cycle in your book. You need to learn the parts of the life cycle. The one that the mosquito is going to infect into the human is called a sporozoite. Okay? They look like a little thing like that. And then when they get into your liver cells, they start to divide mitotically. Okay? And that is called merozoites. And then the merozoites break out of your liver and they get into red blood cells. Merozoites just look like this, so they're just little compounds. And then they multiply in the cells. And in the red blood cells, it's called a trophozoite, because that's where they start feeding on the blood cells. They live on the hemoglobin in your red blood cells. It's protein, it's nutrients for them, see? The trophozoite in the red blood cell, if this was a red blood cell, the trophozoite looks like this. There's one stage that it forms a ring. See? It forms a little ring in the red blood cell. And this is the trophozoite. This is called the ring stage. And that's usually what they use to determine that you have malaria. Okay? The other thing is when the merozoites get in the red blood cell looks like this, and they multiply. Okay? When this thing is done feeding, it starts to divide, and it makes more merozoites. So you'll see some cells with a bunch of dots in it. Sometimes you won't even see the blood cell, it's just all dots. Okay? And so what happens is when they feed, they get their fill, they divide, they produce lots and lots of more merozoites, then they break out of the blood cell, and they infect another red blood cell. See? And they keep infecting more red blood cells, more red blood cells. Every time they break out, your body says, oh my gosh, I'm infected, and then you send out those antibodies and stuff to try to, and they go, quick, send the temperature up. because that's one of your defenses, okay? What do we need? 103? Let's go for 104. We'll take a chance. Maybe 106? No, we might kill the patient. Okay, 104. And then, oh, I think you got them. They're gone. You sweat. You build up the numbers, the feet, build up the numbers, you make more red blood cells. And then, all of a sudden, they all break out at once. You go, boom, the temperature goes up. Quick, I think we're infected. Let's kill them. And you do this cycle over and over again. Each time it gets worse. Each time it gets worse. Some cycles go every 72 hours. Some cycles go every 24 hours. Some cycles go every 48 hours. That's usually specific to the species of malaria. Okay? And so finally you die. You get killed. Sure. It was a lot of work fighting that thing. It's better dead. Fight. So, we don't have many drugs that are very good at killing the parasite because it's in the red blood cell. That's the problem. The second problem is that they have a trick that they use. They change the body codes when they get out. So your body still don't see them all. They bypass them. So the difficulty of working with malaria is trying to look at the disease itself. Tricks. The book gives you some good information on it. Textbook. So we attack the mosquito. We either prevent the mosquito from biting someone, or we kill the mosquitoes. That's our defenses that we normally use. Now, humans have evolved a defense. It's called sickle cell anemia. When your hemoglobin is of a kind that the parasite does not want to eat, it's not going to infect that blood cell. Okay? If you have a severe case, the homozygous situation of sickle cell anemia, you'll live a short life and then die. Okay? If you have a heterozygous carrier, then you live a normal life. And you're also protected. So, do the math. If you have a population, you take no measures at all. You take that population. They are not sickle cell anemia. In time, that entire population will die. No matter how old you are. If you have sickle cell anemia working in the population, 50% of your population will survive. That's a survival tactic. Survival of the fittest, evolution. How did it work? Well, you took one tiny little base pair and you switched it for another base pair, and that made another amino acid. That made hemoglobin with different bonding properties, and under low oxygen tensions, when you don't load up the iron, the hemoglobin changes its shape and changes the shape of the blood cell. One base pair protects one from malaria. Evolution has been accomplished with one base pair change. Can you imagine that? That's how important evolution is. Okay? So that's why I put my atlases out so you can see we have slides of malaria. Okay? Right here. So you want to look for the ring stages, you want to look for the merozoites. Over here is a euglenozoa. Okay? And someone from the last lab did not take the oil off your slide. I don't know who did it. I'm not going to go around and interrogate you. But one of you were naughty and not conscientious. So don't do this. Okay? Take your oil off the slide. I can imagine what your scope looks like, but I won't go there. So, you want to find the trypanosoma. Okay? You found them already. So you already know it. If you already have a drawing of one and you're satisfied with that, then just go with it. Okay? But that belongs to the euglenozoa phylum. And this is also a blood parasite, but this causes sleeping sickness, also a deadly disease. These do not go inside the red blood cell. They stay on the outside, and they consume material in your blood. Okay, and they challenge the oxygen content, so you get very tired. And so you're in the back of class, and 30 minutes into your lecture, you start to fall asleep. Okay, someday you won't wake up. Okay, someone will say, we have an extra student in our class. Oh, we'll call the morgue. Oh, they didn't wake up. My lecture was so boring that this one didn't wake up. Anyway, that's a sleeping sickness that it causes. Now, it's outside the red blood cells, so these things are relying heavily on being able to, every time they divide a new outer cell membrane with different proteins. And so they now cannot be recognized by your body. Every time they divide, they produce a whole generation of non-recognizable members. So they're changing their outer coat all the time. Okay, so that's another one, very hard to fight. Okay, and then if you have time left over, you can, I think we started putting in some diatoms, which belong to the algae family, the algae phylum, which is Chrysophyta. There's some slides out there. You've already done some stuff, but now you can see some more. And are there dinoflagellates out there too? Yeah, so the dinoflagellates is another phylum, Dinoflagellata. Those are really for next time. But if you have, nothing to do, you can get a jump on. Yeah.
So it's a two-part lab, or?
Yeah, it's a two-part lab. Does everyone know how to get your syllabus? Anyone need some help with getting online or getting your syllabus? I think everyone's online because they're grading your papers. So you make sure you check your syllabus. It will also tell you when the field trips are, so you can plan ahead and make your arrangements for the field trips. Okay? Questions? I'll get some more plates, so you don't have to do any of these in any particular order.
Make people feel bad. Yeah, I thought.